So last week we left off talking about genetics and some of the genetic abnormalities that we contend with as a species. And there's a way for us to figure out both probabilities and realities in our children, right? So if we want to know what the likelihood of our child is having or inheriting some sort of genetic disorder, what we can do is we can have ourselves check for whether or not we either express or carry that disorder, and then our mate, whether it's husband, wife, partner, whatever, sperm donor, egg donor, whether or not they carry or express that, and then figure out what the probability of the offspring is having that actual disorder, right? So that's called genetic screening. Historically, we couldn't do that because we didn't have some of the genetic markers that currently we have. And so we applied a few other techniques that we'll talk about in just a few minutes. But generally, these were applied to really high-risk pregnancies. That is, pregnancies where there was a family history of some given genetic disorder, or if the age of the mother was, she was pretty old, like after the age of 35. And even today, women who are having children after the age of 35-ish are considered high-risk pregnancies. Do you guys remember one of the genetic disorders that we talked about that had a positive correlation with the age of the mother?
Down syndrome?
Yeah, Down syndrome, trisomy 21. That's a big one. Now, interestingly, specifically with trisomy 21, there are a number of ways that this can be diagnosed. So, with one of our children, and none of our children ended up having significant genetic disorders, at least none that are diagnosed. With one of ours, in the ultrasound, she had her hands clenched into fists. And the doctor was really concerned about that, and apparently that's like an indication of trisomy 21. So if a fetus, through successive ultrasounds, has his or her fists clenched, that could potentially be trisomy 21. In subsequent ultrasounds, she didn't, so it wasn't a big deal. But anyways, there are a number of ways that we can assess genetic disorders. So historically, one of the things that we used to do is called amniocentesis. And amniocentesis actually extracts fluid from right around the fetus, so the amniotic fluid. And the goal there is to take some of the cells that have been sloughed off of the fetus and then grow them in culture and look for genetic abnormalities. So with amniocentesis, what happens generally is a long syringe is inserted through the mother and through the uterine wall, through the navel. So through your belly button, they stick this long needle and they go in through the uterus and into where the developing fetus is, and they pull some fluids out. It's fairly invasive, not terribly comfortable, so I've been told. Alternatively, there's something called chronic villus sampling. And with chronic villus sampling, instead what they do is they actually take some of the tissue from the placenta, because the placental tissues and the fetal tissues are the same. And so you can karyotype placental tissues, and it gives you the karyotype of the developing fetus. This is a lot less invasive. Generally, they go through the cervix and scrape off placental tissues. And then from that, they can karyotype it and check for specific genetic markers to tell whether or not the fetus is contending with some particular genetic disorder. Additionally, what they can do is they can look for the activity of specific enzymes, because specific enzymes are also correlated with the presence or absence of given genetic disorders. And finally, we can use that technique called PCR and amplify the DNA that we extract from placental tissues, and we can check for specific genetic markers for specific genetic disorders. Yeah, that's right.
So I've heard that some women go in early to get like their DNA tested to see how can they tell how like children, if they have like Down syndrome or anything like that, out of a DNA sample.
So they can't necessarily tell whether or not a child will have some given genetic disorder. They can give you a probability, right? So if you were tested and then the guy that you're having some child with, whether it's, however that's going to be accomplished, if you both are, let's say, heterozygous for some given genetic disorder, let's say it's Tay-Sachs disease, which is a genetic recessive disorder. So we have capital S for normal, which is what both you and this guy are expressing, but you both end up carrying it, right? So if you know this is your genotype for both of you, you can, from that genotype, figure out what the probability is that one of your children will have Tay-Sachs disease. Or if you only plan to have one child, what the probability of that child is of having Tay-Sachs disease. And if it's a genetically recessive disorder, what's the probability that this child will have Tay-Sachs disease? 25%, right? Because in order to actually express the disorder, you have to be homozygous recessive. So if this is the cross, this individual will neither carry nor will it express. This individual won't express, but it will carry, same with this one. But this one will actually express the disorder, right? If one of the parents didn't carry, if they were homozygous dominant, what's the probability that one of the offspring will have Tay-Sachs disease? 0, right? And so by having yourself genetically screened, you can figure out probabilities that your offspring will have specific genetic disorders.
So they can't tell you like, oh, your child will have this, like if you go into the doctor.
No, they can tell you that there's a very high probability that,
Okay.
unless it's a dominant disorder, right? So you're clearly under the age of 35, which means that you wouldn't express Huntington's disease if you carried it. But you could go in and they could say, oh yes, you carry the dominant character, the dominant traits, so your child will have Huntington's disease at some point. Okay. So finally, what we can do, and this is really what you were talking about, Sarai, is we can have ourselves genetically screened. We can go in and have ourselves, our own genomes, assessed to figure out what the probability is that we're going to carry specific genetic disorders. And typically what we're looking for are single nucleotide polymorphisms, is what they're called, or SNPs. And that's where we're looking for some single nucleotide amid a gene sequence that indicates that there is some specific disorder. Also, with that process of in vitro fertilization, this is what we were talking about when we talked about embryonic stem cells. With in vitro fertilization, what the doctors can do is they can karyotype eggs, and they can make sure that these eggs that are going to be implanted in the recipient mother don't carry any of the genetic disorders that you don't want your child to contend with. Okay. Questions about genetic screening? Questions about genetics in general? Okay. Oh yes, Linda.
What does SNP stand for again?
Single nucleotide polymorphisms. Okay. So what we're going to do now then is shift our attention from genetics over to DNA.